Dear student, welcome to our program, Knowledge Quest. The subject is biology. In our previous lesson, we were discussing the process of cell division so that we get to understand why we say a parent cell has 46 chromosomes while a daughter cell has 23 chromosomes. And for that reason, we want to continue from where we stopped. We said that during gamete formation, gametes, they are formed during the process of cell, during the process of meiosis. We said that gametes are formed during the process of meiosis. And of course, when we talk about meiosis, it's a type of cell division. We have two types of cell division, whereby we talk about mitosis and meiosis. Now, in meiosis, we said that meiosis takes place in two phases. We have the first phase, we call Meiosis I, and we have the second phase, we call Meiosis II. Now, we said that during Meiosis I, the homologous chromosomes, they separate. And we said that during that process, there are 5 phases through which the cell undergoes so as to separate the homologous chromosomes. And we said that homologous chromosomes, these are chromosomes that appear to be similar. They appear same, but they may be having the same genetic material, or they may be having different genetic material. And for that reason, we said that there are 5 phases. We said the first phase is interphase. We have the second phase we called prophase. We said the third phase is metaphase. The fourth phase is anaphase. And the last phase is telophase. And because we are discussing the first meiotic division, then we said that we talk about interphase I, prophase I, metaphase I, anaphase I, and telophase I. Now, briefly, we discussed about the first two. We discussed about interphase. We said that interphase is a phase of cell preparation. The cell has to prepare itself to undergo the whole process of cell division. And we were able to mention some of the activities that takes place during interphase. We said that the cell builds up enough energy for the whole process, and we said that that energy is stored in form of adenosine triphosphate, that is ATP. We also said that the genetic material multiply. The cell organelles, they also multiply. Then we went ahead and discussed the prophase. We said that during the prophase, you realize that the nucleolus disappear, the nuclear membrane starts to disintegrate, and then we said that the centrioles, remember the first phase, the cell organelles had duplicated. So we expect even the centrioles to be true. We say that the centrioles, they move towards the opposite poles. So we discussed the first two. And maybe I can just draw the cell at that point so that we are able to follow. So we say during interphase I, we said that an animal cell does not have a definite shape, but I decided to use that shape. Then we said at this point, the chromosomes, they appear as thin strands. They are not visible. You cannot see the chromatids, but at least you can see the chromosomes. At this point, we say that we have the centriole, we have the cytoplasm, which suspends all the cell organelles. And then we said this is the cell membrane. We have the cell membrane. We said we have the cytoplasm. Then, of course, we said this is the nuclear membrane. And then we have the centriole. And the role of the centriole is to form the spindle fibers. Then inside the nucleus, here where we have the nucleus, we said that we have the homologous chromosome. And then I said I'm using different shades that just to show that these are two different chromosomes. It's only that they appear to be the same. So that is the cell, that is the shape of the cell, or that is how the cell appears during interphase 1. And we say that at this point, you are not able to see the chromosomes clearly because they are just thin strands. Then we say that during the prophase 1, we have the cell at this point. We said a cell, animal cell does not have a definite shape. We said that the nuclear membrane begins to disintegrate, meaning at the end of prophase 1, we don't expect to have the nuclear membrane. And then we say that the nucleolus, inside here we have the nucleolus, and we say that during interphase you are able to see the nucleolus, but during prophase 1, the nucleolus disappears. And then we said the spindle fibers, they start forming from the centrioles. And we said the centrioles, they move towards the opposite poles. And then we see the spindle fibers start forming. Remember the spindle fibers, at that point, they are going to play a role of separation. Then we said at this point, during prophase, the chromosomes now, they are clear, they are visible. You can see them. Why are we able to see them at this point? You realize that at that point, the chromosomes, they shorten. In the process of shortening, they thicken. So as a result, you are able to see the chromosome having the two chromatids. There is something that happens during prophase 1. During prophase 1, the homologous chromosomes, yes, they are visible. We can see them. What happens? They lie close to each other to a point you realize that the chromatids may lie on top of the other. So in a way, I want to show that at one point, they are going to, so if I can draw it here, that is during prophase. During prophase 1, you have two chromosomes, and these two chromosomes, at one point, they tend to lie close to each other. So you see, this is one chromosome, this is another chromosome, but at one point, the chromatids, they are lying close to each other to a point whereby they are attached. So the point where they are attached here, we call it chiasma. That point of attachment between the two homologous chromosomes. Remember, these are homologous chromosomes, meaning they appear to be the same, but may be of same genetic material, but or different genetic material. So we are saying that at this point, during prophase 1, the homologous chromosomes, they are lying close to each other. So in a way, they form what we call a bivalent. Bi means two, bivalent. So we refer now to these two as bivalent. They pair up. In the process, remember I'm only using two chromosomes, but we don't have two chromosomes. We have 46 chromosomes. So I've only picked two chromosomes just for the sake of the learning for this lesson. So we are saying that during prophase 1, something is happening. The two homologous chromosomes lie close to each other to an extent of joining or connecting at a point we call the chiasma. This is the point whereby crossing over takes place. Crossing over. Now, what do we refer to when we say crossing over? It means that this chromatid is carrying specific genes, probably which are different from this chromosome. We know that each chromosome carries different genes. There are chromosomes, there are genes for maybe height, there are genes for skin color, there are genes for sex. There are different genes which are carried at a given chromosome. So you realize that because of lying close to each other, there might be high chances of exchanging the genetic makeup, such that what is in the chromosome 1 at this point will move to what is chromosome 2 at this point. Then the genes, so we are saying that if you were to draw this, then we are going to have something like this. So we are saying you have this chromosome. Meaning, at this point, there was exchange of genetic material. So what happened? The genes on this chromatid, they moved to this part. And the genes for this other chromatid moved to this chromatid. So if you were to check, you realize that this chromatid will not be the same as the original chromatid that we had. Meaning, there is exchange of genes that has taken place at the point of the chiasma. And that is what we refer to as crossing over. So basically, we are saying that during prophase 1, what is happening? The nuclear membrane begins to disintegrate. Number 2, the nucleolus disappear. Number 3, the homologous chromosomes lie side by side, getting to a point of connection at the chiasma. We said at this point they are going to form the chiasma. Then number 4, we said that the centrioles, they move towards the opposite poles, and the spindle fibers, they begin to form. Then that marks the end of prophase 1. So where does crossing over takes place? So crossing over takes place in prophase 1. And I've said crossing over is whereby the genes from one chromatid is exchanging with the genes from the other chromatid. So if you have the two chromatids, they will not be of the same characteristic. That's why you can have brothers born by the same father, but they don't look similar. During meiosis, during cell division, during prophase 1, crossing over takes place. So the gametes that will be formed during when one son is born and the other one will be different. So we'll continue to the next phase, whereby we say that from the interphase, we move to prophase, and then we move to metaphase. So during metaphase, this is, we say, metaphase 1, because we are still in the first meiotic division. Now, something happens. Remember we had said in prophase 1, the nuclear membrane had started to disintegrate. So what does that mean? At the end of prophase, or in the beginning of metaphase 1, we don't expect to have the nuclear membrane. And if we don't have the nuclear membrane, it means the whole contents of the nucleus is now suspended in the cytoplasm. So if I have to draw that, I will say this is the cytoplasm. We have the centrioles at the opposite poles. And what happens, because the homologous chromosomes, they are now suspended in the cytoplasm. So during metaphase 1, these homologous chromosomes, they align themselves at the equator of the spindle fibers. So if I was to draw the spindle fibers, we say that the centrioles, they form the spindle fibers that are necessary during cell division. So what do we say? We say that the homologous chromosomes, they will align at the equator, that is at the center of the spindle fibers. Then they will be held at the centromere by the spindle fiber. So if I have to draw this, then I will say I have this one. Then we also have another one here. Remember, these are bivalent chromosomes. They are bivalent chromosomes. They are homologous chromosomes. And don't forget that they are connected. The two homologous chromosomes, they were connected at the chiasma. At the same time, don't forget that these chromosomes, they are held by the spindle fibers at the centromere. So we expect the spindle fibers to hold the chromosome at the center, that is at the centromere. So this is where we have the centromere. So the spindle fibers will attach the chromosomes at the centromere. But at the same time, don't forget that we have the chiasma where the two homologous chromosomes are connected. That is what happened during prophase 1. So basically, what are we saying? We are saying, number one, during metaphase 1, the homologous chromosomes, they align at the equator of the spindle fibers. Remember, the centrioles had already moved towards the opposite poles. Then at this point, we say that the spindle fibers, they hold the chromosomes at the centromere. That is the end of metaphase. Then what do we expect in the next phase? So in the next phase, we have anaphase. Anaphase 1. Now, we are learning about cell division, and we are saying it is a whole process. And so far, we have not seen where the division is taking place. And that is why anaphase, I call it the phase of cell division. This is where cell division takes place. Anaphase, I call it the phase of cell division. All these others were just the processes. Now, why do I say so? It is because during anaphase, something happens. The spindle fibers, remember they were formed from the centrioles. The spindle fibers, they start to shorten. Now, if this is a spindle fiber, this is a centriole. From the centriole, I'm forming the spindle fiber. Then from another centriole, I'm forming another spindle fiber. Then, of course, at this point, they are holding something. Let me say they are holding something at this point. So these ones are holding something here, and these ones are holding something here. Now, what do you think will happen when this spindle fiber starts to shorten? definitely when I say to shorten, it means they will start taking this direction. So they are pulling back to the what? To the centriole. So they are pulling back to the centriole. So when they shorten, they take this direction. Remember, the spindle fibers had formed, so they were holding the two. So when they shorten, it means they pull these two away from each other, assuming they were attached together. So basically, that's what happens that during anaphase, the spindle fiber that are holding the chromosomes, that is at the centromere, they start shortening. So in the process of shortening, they pull away, they separate the two homologous chromosomes. Remember we said during the first meiotic division, we are separating the homologous chromosome. So at what point are we able to see the separation? It is at the anaphase 1. So if I was to draw, then I will say, I assume I have this cell. Then I have the spindle fiber on the other side. The spindle fibers, they are holding. Remember, these are the chromosomes. They were homologous chromosomes. But don't forget that something happened. Don't forget that something happened. So at this point, don't forget that we have a different form of gene because of the crossing over. So I will also draw another one here. Then at this point, remember there was crossing over. So we also expect another set of chromatid to be having some genes from this chromosome. And this set of chromatid is also having some genes from the other chromosome. And remember, they were held by the spindle fiber. So they were held by the spindle fiber. And we are saying that the spindle fibers, they are shortening. So in the process of the spindle fibers shortening, they are pulling the homologous chromosomes away from each other. So in a way, the homologous chromosomes have been separated and they move towards the opposite poles. Now, what happens towards the end of anaphase? Towards the end of anaphase, you realize that immediately this chromosome is going to be enclosed by the nuclear membrane. The other chromosome is going to be enclosed by the nuclear membrane. Then, immediately, we see the nucleolus reappearing. Let me try to show that. So, if this is the cell, so at this point, whereby we have this chromosome, we have this chromosome. So immediately we see the nuclear membrane appearing. So the nuclear membrane encloses this chromosome. The same case happens to this other one. So here, remember, we had crossing over. So the same case, we see the nuclear membrane forming around the chromosome. Then, remember, we have our centriole. Remember the spindle fibers, they shortened. We also have another centriole. At this point, we don't forget that we have the cytoplasm. Don't forget that we have the cytoplasm. So we are seeing that the nucleolus is reappearing towards the anaphase. So at this point, then I expect to show the nucleolus. I also show the nucleolus this side. Then what happens? Now we move to the last phase. We call it telophase. We call it telophase. That is telophase I. Now, telophase I, something happens. During telophase I, something happens. Hoping that you've been following where we started from. Interphase I, prophase I, metaphase I, and anaphase I. And if you are very keen, I want you to start imagining on this cell what will happen if it will be of the same with the first one we started with. But before I continue to the next phase, which is telophase I, let's go for a short break. Stay tuned. We'll be back. Dear student, welcome back to our program, Knowledge Quest. Before we went for a short break, we were discussing the process of meiosis I, which results to the formation of gamete cells. And we said that gamete cells, they have the number of chromosomes as the parent cell. Now, we discussed interphase I. So far, we have discussed up to anaphase I. And we say that during anaphase I, I call it, it's a point of cell division. We are saying cell division is a whole it's a process, but if you are very keen, you move from interphase I all the way to metaphase I. We have not witnessed any division. So the division is taking place at the anaphase I. And I said that during anaphase I, the centrioles, which formed the spindle fibers, so the spindle fibers, they start to shorten. In the process of shortening, they pull away the homologous chromosomes away from each other. And as a result, they cause the cell division. Then we said, towards the end of anaphase I, we expect the chromosomes to be enclosed by the cell membrane. This is the cell membrane. Cell membrane. And then we say that the nucleolus also reappear. The last phase now, telophase I, that is in meiotic I, the last phase is telophase I. During telophase I, something happens. The cytoplasm divides. If you remember very keen, we said that cell division starts by the division of the nucleus, which we have seen the nucleus dividing. Remember where we started? And so far, we have two. The nucleus divides first, and then followed by the cytoplasm dividing. So you want to see how does the cytoplasm divide. Now what happens is that this cell that is at telophase I, the cell membrane undergoes what we call constriction. So it undergoes what we call constriction. Okay? This cell, the cell membrane undergoes what we call constriction. Now, when it constricts, something happens. Remember here, we have our centriole. Remember here, we have our nucleus. And in the nucleus, we have our chromosome. And remember this chromosome, it has what? A different portion because of crossing over. Don't forget that the cell organelle had duplicated from the beginning. So when the cytoplasm is dividing, then it means everything is dividing into two. So the same case here, we are going to have another cell with the nucleolus. We say that the nucleolus reappear. Then we have this one. But at one point, remember that there was crossing over. Then there is also the centriole. But now, what do we expect? This part constricts and then it divides into two. So when this part constricts and divides into two, what do we see? We end up having two cells. We have two cells, and these two cells, they are the same. So we are going to have two cells, one cell here and another cell here. And this cell has the what? The chromosome. That is the centriole. Then the chromosome that we have towards the telophase, the moment we form the two cells, what happens? This chromosome, it now becomes very thin. So we see it as very thin, so you may not be able to see clearly that it is what? It's a chromosome. Remember, it's a continuous process. So at this point, we are going to start again another process, which is meiosis II. So again, at this point, we also have another chromosome. But remember that at one point, there was exchange of genetic material. At one point, we exchanged the genetic material. Then this cell now, there is the nucleolus. We also have the nucleolus. We have the centriole. So if you compare on this cell, it should be the same as that. If you compare on this cell, it should be the same as this one. It's only that at this point, the chromosome may appear as thin strand. But this, the number of chromosomes, they will be 2. It's only that we have the chromosome here. Here we have homologous chromosome. Here we just have one chromosome. Here we have 2. Here we have 1. But we are saying that at the end of it, we are going to see they are going to look like thin strands. So if you will see them, you will see them as thin strands. They will be observed as thin strands. So basically, what are we saying? We are saying that during meiosis I, we are separating homologous chromosome. And homologous chromosomes, at that point whereby we are having telophase I, at the end of it, the homologous chromosome have separated, so we have the chromosome. But this chromosome, crossing over had taken place, so the genes at one point, they are different from the other. Now, meiosis I and meiosis II, it's a process. We expect the moment the cell starts the process of meiosis I, we expect at the end of meiosis I, the process to continue to meiosis II. Although the cell may undergo a bit of rest, but we expect the whole process to be continuous. And because of that, let's see what happens during meiosis II. So in this case, we are discussing meiosis II. And meiosis II, In this case, we are going to separate. We are separating. That is, in meiosis II, we are separating sister chromatid. Remember, I'm saying sister chromatid because we said a chromosome is made up of 2 chromatids. And so far, we had 2 chromosomes that looked similar, but we have separated them. So we have separated the homologous chromosome. So we move on to separate the sister chromatid. Remember, we are saying at the end of meiosis II, now we are going to form 4 daughter cells. And these daughter cells, each of them is going to have the number of chromosomes as the parent. But during meiosis I, the 2 daughter cells each had the same number of chromosomes as the parent. So let's see what happens. The same phases takes place. So we talk about interphase II. The other time, we said that interphase is a phase of cell preparation. And now this time I want to say that during interphase II, the cell may go through interphase II or may not. Why? During interphase, remember we were saying there was preparation. Which preparation did we do? The genetic material was multiplied in the first interphase. Energy was built up in the first interphase. So basically, what we are saying that the cell might be just undergoing rest at this point, but it is good we draw that cell. It is good we draw the cell so that we see this is the cell. And at this time, remember, there are 2 cells. There were 2 cells, and each of them is going to go through the same process. So I'll pick 1 cell, but each cell will go the same process. And this cell, remember, we have a homologous chromosome, which separated to give us the chromosome now. So that is what we have. Remember, the nucleolus is there because it reappeared during the anaphase. Then we have our cytoplasm. We have our cell membrane. This is the cell membrane. We have our cytoplasm. We have our centriole. Although now at this point, the only thing that we may expect to happen is the centriole to replicate, because now at this point we have 1 centriole. But all other processes took place during the first interphase. But the centriole may replicate so that at least during prophase we have 2 centrioles because we need the 2 centrioles. So at this point you see the chromosome may not be that visible, but it is 1. The other time there were 2. This time it is 1. And if you are very keen, there is a difference here. Now let's see what happens during prophase. That is during prophase II. During prophase II, nucleolus again disappears. Remember here it reappeared during anaphase. That is during anaphase I. That is where we were able to divide the nucleus, and then now it was enclosed. The chromosome was enclosed by the nucleus, and we formed a cell. So meaning at this point, our cell is having the nucleolus, which had disappeared the first prophase I, but reappeared at anaphase I. So we have the nucleolus. So during prophase II, number one, we want to assume that the centriole at this point has duplicated. So we have two centriole. So the centriole will move towards the opposite poles. So they are true. The spindle fibers will start forming. So it's like the same process that is repeating. But the only thing that is we are separating the sister chromatid. The nuclear membrane also begins to disintegrate such that at the end of the prophase II, you realize that we don't have the nucleus. So that is the prophase. So at this point, the nuclear membrane begins to disintegrate. Nucleolus disappears, so we don't have it. The centrioles, they move towards the opposite poles. Then the spindle fibers, they start forming. Then at this point, remember we have our chromosome. The chromosome is very visible. Why? During prophase, the chromosomes, they thicken and shorten. And as a result of that, they are visible. So we are going to draw and make it more visible. But at this point, remember there was crossing over. So this portion of this chromatid has a portion of different genes that was not there from the initial because of the crossing over. Then we have the cytoplasm. So that is the beginning of prophase II. But we expect towards the end of prophase II, the centrioles, they have moved towards the opposite pole. The nuclear membrane completely has disintegrated, such that now we move to metaphase II. Now, the key word during metaphase II or during metaphase I, usually the chromosomes, they align at the equator of the spindle fibers. Why do they align at the equator? Remember, they are suspended in the cytoplasm. Why? The nuclear membrane has already disintegrated, so they are just in the cytoplasm. So if I draw this, then I'm going to have, so our chromosome is just at the equator. And it is, remember, we cannot forget our portion. Then it is held at the centromere by the spindle fiber. Remember, the spindle fiber, they originate from the centriole. This is the centriole. This is the cytoplasm. So we have the cytoplasm. So we are saying the key word or the key process that is taking place during metaphase II is that the chromosomes, they align at the equator. Then they are held by the spindle fibers at the centromere. While they are at the equator, you realize that, number one, they are not now confined in the nuclear membrane. The nuclear membrane disintegrated during prophase II. So at metaphase II, we don't have the nucleus. Number two, we don't have the nucleolus. But of course, all other cell organelles are present. All other cell organelles, they are present. Now, what happens? We move to the next phase, anaphase. Anaphase II. And as a student, I said, there are key things that you need to note. What happens at this phase? What happens at this phase? Division, cell division always takes place at the anaphase. Whether it is anaphase I, whether it is anaphase II, always cell division takes place at the anaphase. So, and what causes, you also need to understand what causes this division to take place. Cell division. You we need to understand what causes the cell division to take place. Because always at anaphase, the spindle fibers which are holding the chromosome at the centromere, they always start to shorten. And definitely when they shorten, they move away from each other. So they go back to the central. In the process, they separate. In the process, they separate. So we say, at this point, we are going to have the spindle fibers shortening. So in the process of shortening, they are picking the what? They are separating the sister chromatid. Remember, they were held at the centromere. We also have another one here. But this one, remember, a portion of it is having different genes. So it's also held at the centromere by the spindle fiber. So we are saying, in a way, the spindle fibers, they are separating. So if I can show that point of separation, then we can say they are pulling away from each other. So the spindle fibers are moving towards the centrioles, and the centrioles are found at the opposite poles. Now, towards the end of Anaphase II, something happens. The chromosomes, they reach at the opposite poles. And when they reach at the opposite poles, immediately they are enclosed. This chromatid, it was a chromosome. Now it has separated. It is a chromatid. This is one chromatid, this is another chromatid. So that chromatid, immediately it will be enclosed by the nucleus. And that point of enclosed by the nucleus, we no longer now call it a chromatid. It becomes another chromosome. Immediately, the nucleolus reappears. Now, let's just try to see what is happening. So let me move it here. Anaphase II. So we are saying at Anaphase II, so that we are able to see Telophase II. Telophase II. So at Telophase II, there is always a constriction of the cell. But here we said that that nucleus, the nucleus will enclose. It will enclose the chromatid. At that point, it will be seen as a thin strand. Then we have our centriole. Then the nucleolus will reappear. So maybe let me show it to be. Then we also have another one here. So this is at the end of Telophase II. Then we also have another one here. But remember, one of it has different genes. Then we have our nucleolus, we have the centriole, we have the cytoplasm. So at this point, that is towards the end. Of course, I explained and drew what happened at the beginning of Anaphase II. So towards the end, the chromosome has moved towards the opposite poles, and then it has been enclosed by the nuclear membrane. Then something happens. So at this point, we have the chromosome. Then we have the centriole. We also have another one here. So there is the centromere. We have the nucleolus. Nucleolus reappear. It reappeared towards the end of Anaphase II. Then we have our centriole. We can't miss the cytoplasm here at this point. So during Telophase II, there is a constriction that results to the separation of the two cells. Now, at the end of Anaphase I, we expect that the sister chromatids have separated. They have moved towards the opposite poles. And at that point, we may not call them the chromatid, but of course, we'll call it the chromosome. But towards the end of Anaphase, we expect at the end of Anaphase II or early Telophase II, we expect that chromatid to be enclosed by the nucleus. Then we expect the nucleolus to reappear. After the nucleolus reappear, we expect the two to be enclosed by the nuclear membrane, so that now we are going to have a nucleus. Then we are having the centrioles. So at both ends, we can see the centrioles. Now what happens is that at Telophase II, remember we are now marking the end of meiosis. We started from Meiosis I to Meiosis II. At the end of Telophase II, we now have a cell. We have a nucleus. We have a cell that has constricted. So we say the cell constricts. The cell membrane constricts. That is the cytoplasm. We want to see how the cytoplasm will divide. Then at this point, we have this chromosome. Remember, there was crossing over, so this one is having different genes. Then we have the nucleolus. Our central is here. We have the cytoplasm. Then we also have another chromatid. We also have the nucleolus and the centriole. So basically what happens during Telophase II? So during Telophase II, we are saying that the chromosome that had separated, they move towards the opposite poles. They are enclosed by the nucleus. The nucleolus reappear at that point. Let me not draw it so that it looks the same. Let me say that at this point, we only have, so that you don't confuse between this, because it is towards the end we expect some of these processes to take place. So we expect at this point, so if this is the cytoplasm, we have the centriole. So at anaphase, we said that the sister chromatids have moved towards the opposite poles. And then, so such that now at telophase, we are seeing that at telophase, number one, the chromatid is enclosed by the nucleus. Number two, the nucleolus reappear. Number three, the cell membrane or the cytoplasm, the cell membrane constricts. And why is it constricting? So that we can divide the cytoplasm. Remember we said cell division starts by dividing the nucleus, then we divide the cytoplasm. So at the end of the day, what we have, we are going to have two daughter cells. Remember we used one cell, but we said the two cells, each was dividing individually at the same time, of course. So if I am to draw the outcome of these cells, let me draw them up here. We are going to have two cells. Remember we said an animal cell does not have a definite shape. It is the teacher who has decided to use that shape for the sake of explaining. An animal cell does not have a definite shape. So we are going to have a cell. Inside here we are going to have the chromosome. Remember that we said the moment it is enclosed by the nucleus, we no longer call it the chromatid, but now the chromosome. But it will look as a thin strand. So, for the sake of showing the crossing over that took place, so I'm only going to draw it like this. Then, our centriole, we have it. Our cytoplasm, we have it. And our nucleolus had reappeared, so we expect it to be present. Then the other cell, we are going to have another chromatid, but remember, one of the chromatid has different genetic makeup. Then we have the nucleolus, we have the centriole, then the cytoplasm. Remember, we had two cells, two daughter cells at the end of meiosis I. One daughter cell has undergone cell division to form two daughter cells. Definitely, the other daughter cell will also undergo the same cell division to form again two daughter cells. So I'm also going to draw the other. So remember, we are saying that both of them are undergoing cell division. It's only that I picked one to explain. So we assume that is the nucleus. Then we have the centriole. Now, at this point, don't forget that the other one also had another portion of the gene. Then we also have this one. So at the end of the day, how many daughter cells do we have? There are 4. At first, we started by one cell. One cell divided to form two cells. Each of the two cells underwent cell division to form again two cells. So at the end of the day, we have 4 daughter cells. But each of the daughter cells, they are not of the same. Why? Let me number these cells. Assume this is daughter cell 1. This is daughter cell 2. This is daughter cell 3. This is daughter cell 4. And remember what we said, that during crossing over, something had happened. Remember also what we said, that we started by homologous chromosomes. And these homologous chromosomes, we said they only looked similar. They appeared similar, but they were not, they may be having the same genetic makeup or different genetic makeup. So what does that mean? They may be having the same genetic makeup or different. Then, at the end of the day, again, we are seeing crossing over taking place. So far we have 4 daughter cells. So look at daughter cell number 1, daughter cell number 2, daughter cell number 3, daughter cell number 4. All of them, we say they have a half the number of chromosomes as the parent, because the parent had 46. But now, each of them has a half, meaning each of them has 23 chromosomes. So if you check on the genetic makeup of this daughter cell, this daughter cell, this daughter cell, definitely they are totally different. But these cells were formed by the same person. That's why you find that during fertilization, maybe it is this cell or this gamete that fused with another one. Or during another fertilization, it was this cell that fused with another cell. During another fertilization, so assuming this is the father, assuming it is the father, so these are sperm cells. So maybe the first time it was this sperm that fertilized the ovum. The second time it was this. So the chances, it's a chance during fertilization. So you find that children are of the same. They are of the same parent, but they may look different. Why are they looking different? They're crossing over. The genes exchanged. So basically, if you have been following, I think now you've understood how the 46 chromosomes in a parent, that is in body cells, they are being halved. They are halved into 23 chromosomes in a gamete cell. A gamete cell, either a sperm cell or an ovum cell. Now, once you understand that, then in the next lesson, we are going to explain how fertilization now brings about exchange of genetic makeup. And at the end of it, we are going to see why we have variations among organisms of the same species. Thank you for watching. I've been your teacher, Lydia Siago. Our program is Knowledge Quest. Always get tuned.